videos in my office again. So hi. So first thing we're going to talk about is the bacteriophage reproduction. We're going to talk about the lytic reproduction and the lysogenic reproduction. So the lytic reproduction basically, so virions do not have a mode of transportation, so they interact or they come in contact with the bacterium through random chance. So the more virion there are, the higher chance of that random meeting it. Once it comes in contact with the host cell, it attaches. Then once it attaches, it injects its genome into the host cell. And once that host cell enters, that genome enters the host cell, the viral enzyme degrades the host DNA. And then once the host has bacterial DNA has lost its DNA, its chromosome, the host cell's enzyme and ribosomes begin synthesizing the new viral nucleic acids and proteins under the control of the viral genome. This and then it's followed by the assembly of new virions within the host cell. And once those mature virions, mature, they are released out of the host in a process known as lysis. So it bursts open. So pretty horrific sounding, rather alien. Anyway, gross. So the other form of reproduction is the lysogenic reproduction. It's a lot more subtle. So this one refers to the viral cell reproduction, in which the infected host cell doesn't lyse for several generations, and in some cases, quite a long time, many generations before they lyse. So it starts with, so the cycle starts much like the lytic reproduction, a random attachment, random association meeting it, and then it attaches, injects its genome. Only with the lysogenic reproduction, the host's DNA is not degraded. The phage genome doesn't immediately assume control of the cell, as in with lytic reproduction. Once in the host cell, the virus remains inactive, but this inactive bacteriophage is called a prophage. So the prophage codes for a protein that suppresses the prophage gene. So the side effect of this is actually that the bacteria becomes resistant to infection by a similar virus. So the prophage inserts its DNA into the bacterium and becomes a physical part of the bacterial chromosome. So basically its own DNA physically binds with the bacterial DNA and chromosome. So then every time the bacteria replicates, the virus prophage is also replicated. So every daughter cell of that bacteria is infected. Bacteriophages can remain part of the bacterial chromosomes for generations, or forever for that matter, and in fact are responsible for the change in phenotype of bacteria. So this process is called the lyogenic conversion, and it can turn bacteria into pathogenic infections. So bacteriophage genes are responsible for disease-causing toxins and proteins found in bacterial agents like diphtheria and cholera and severe E. coli. So if you have any questions, I look forward to talking to you. Thanks. Bye.